Good morning once again, and welcome to today's webinar, which is titled From Wild Reef to Situ System, Bridging Aquaculture and Conservation. Our speakers today are Gimel Morley, who is our Coral Aquarist Manager here at the Perry Institute, and she will be joined by Stefana Roberts, who is a Coral Aquarist here at the Perry Institute. So just a few housekeeping rules before we begin. A reminder to keep your microphone muted during the session. And also please note that this webinar is being recorded. Afterwards, there will be a Q&A session. So add any questions that you may have into the chat or the Q&A option, or you can raise your hand at the end. Also, please be aware that we will be sharing an audience feedback link in the chat, and we will be very grateful if you fill it out for us. So without further ado, I will now welcome our speakers to begin their presentation.
Good morning, everyone. I hope that you can hear me loud and clear. I am in the gene bank, so that's why my video might be a little bit blue, but I am Gimel Morley, the Coral Aquarist Manager at the Perry Institute for Marine Science. I'm here at the Coral Gene Bank, and today we're just going to go into what it's like working at the gene bank and how we got it started. So for me, my background is in aquaculture and nutrition. I have a master's in it. And I have worked in freshwater tilapia and in saltwater shrimp. And now I work with coral conservation. But my duties here at the Perry Institute at the Coral Gene Bank is to oversee coral husbandry and all of its systems health, to manage coral collections and the quarantine and make sure that everyone is following biosecurity procedures, to supervise staff and interns. Yes, we do have internship opportunities for those tuning in. If you're located in the Bahamas at UB or at BAMSI, feel free to apply. And I'm here to oversee daily operations and to liaison with partners. For today's webinar, we're going to be covering the understanding of the coral reef ecosystems and some of their threats that they're undergoing in our Bahamian waters. The daily operations of the Living Coral Gene Bank, and I'll explain what a Living Coral Gene Bank is. We'll be transitioning from the wild reef to the ex situ systems that we have, and how we got the corals from the wild to our systems, and the role of aquaculture in conservation, in coral conservation, as well as how it benefits the Bahamas and any potential researchers in the Bahamas. So let's start with understanding our reefs. Corals cover less than 0.1% of the ocean floor, but they support 25% of the marine species. They're completely vital for biodiversity, not just for coral biodiversity, but for fish and other invertebrates that are in the marine habitat. We need our corals for ecosystems being able to function and for resilience, for economic importance, such as fisheries, tourism. For the Bahamas, tourism is a vital part for us. We get a lot of our income from that. So protecting our coral reef should be a high priority for us. But our reefs are in trouble and they're undergoing a wide variety of stresses. They are experiencing prolonged cycles of bleaching, ocean warming, various coral diseases such as stony coral tissue loss disease. And with stony coral tissue loss disease, you can see a rapid degeneration of the tissue from our stony corals, which are a major reef building corals. We have other diseases out there in our oceans, but that is the one that our lab predominantly focuses on. We also deal with overfishing disruptions in our corals, pollution, anthropogenic destructions from people dropping their anchors and just dragging them, or persons stepping on the corals, having sunscreen on their skin, and then going into corals. Tourism, yes, it is a vital part of the Bahamas, but that's also contributing to the degradation of our reefs. So all of these stress, they're why corals are actually important and why we need to focus on coral conservation. With this, we need to act fast because we have increasing environmental stressors on our reef, not just from anthropogenic factors, but that disease that I talked about, stony coral tissue loss disease. Whenever we go out to the reef, we see that that disease is claiming a lot of corals, just massively wiping them out. You would go to one reef maybe in 1 month, and then the next month, that entire reef system just might be gone just because of that disease. Some other things might come into play, but stony coral tissue loss disease is playing a major factor in our loss. Coral conservation is important for us because for us, we need to keep up the genetic diversity in our corals and in our reef systems. We don't just want to rely on one reef species. So if we can save as many different corals as possible, then that can help us rebuild our reef. So that's what our lab focuses on. We're trying to preserve as many different colonies of stony corals as possible so that we can have that genetic diversity. So hurricanes are a factor of stress as well. We live in a hurricane zone. We get hit by hurricanes almost every year. And I know a lot of people focus on mangrove restoration, and yes, mangroves, they're wonderful as well at protecting our shorelines, protecting us from erosion, but corals are there too. Corals are there to protect us from all of those wave breaks as well. So it helps us to decrease our storm erosion, our beach erosion, and those storm surges. And implementing various different strategies of coral conservation is what we strive to do here at the Perry Institute. So our facility is classified as a living coral gene bank. And that just means that we collect a lot of live samples of corals from the wild. So not just gametes, we collect living tissue samples of corals. So some whole colonies, some fragments, but all of it, it's living organisms that we're placing in our systems. And within our systems, if you look in my background, you might see some lights overhead. Those are our lights. So those simulate sunshine in our system. We are an intensive aquaculture facility where we create our own seawater using RODI water and then converting that using a salt mix so that we can add sterilized seawater into our systems. We also have our lights on we program them so that they can go through sunrise and sunset simulations as well as moonrise and moonset. So we have lunar cycles and sunrise cycles. And with our water quality, we try to mimic the ocean in our system as much as possible. We don't have fish in there, just putting that out there, but we do make sure that the corals are receiving enough flow and that our water quality is similar to what they would find out in their natural environment. We also feed our corals. So I know many people might think that corals are rocks. They're not. They're living organisms that can photosynthesize like plants, but they also take in food. So some corals facilities, they feed them brine shrimp or mysis shrimp or rotifers or copepods. For us, we try to, we're still figuring out what we want to do. So I'll go into what we feed them later on, but with our coral gene bank, we want to have this facility so that we can support research and restoration practices. So we're holding corals. That's what we do. We just hold them. And we plan to spawn them, and their offsprings, those would be the ones that we plan to replant into our reefs. Also, since we're holding corals, scientists in the Bahamas can potentially even use them as experiments in the future. They would have to go through UB, but it's a great opportunity for collaborations. So we're here, we're letting the corals grow and seeing what they do in our tanks. And then you guys who live here and our scientists can potentially come and see our gene bank. We are a biosecure facility, so it might take a little bit of time to get approval and for you to apply to come here, and you do have to follow certain procedures in order to get here. We're not open to the public, but we're here for persons in the scientific community to come in and view and see what we're doing. So on each one of our systems, this is what we have. It's not all of the equipment that we use, but it's some of them. So we have tanks and stands that are made out of fiberglass. So one of the coral aquarists, he couldn't make it today, but he left some notes for us, just for us to share. So the tanks behind us are made out of fiberglass, and they were specifically configured just for us. And all of our systems roughly hold about 800 gallons of water flowing through them. Our pumps run about 400 gallons per hour. And this is a closed loop system. So we have 3 holding tanks and 1 sump tank. I'll show a picture of this afterwards and videos. But it's 3 holding tanks to 1 sump tank. And then we have 3 holding systems that way. And each system roughly holds like I said, each system holds about 800 gallons through it. Our sump alone holds about 180 gallons, and we have live rock in our sump. And that way we're able to have bacteria so that we can help the corals take some of those nitrogen particles and go through the nitrogen cycle. So converting that ammonia to nitrite, then nitrate, just so that it can be in a less harmful form for the corals to live in. Since we don't have any fish in our systems, that just means that we have less amounts of nitrogen in the system, but we feed the corals. So that adds to the amount of ammonia that's in the tank. We have apex controls that control our light and for monitoring the temperature and pH. We have titanium heaters and, well, 2 titanium heaters on each one of the systems. And these keep the temperatures in the water at a specific temperature range. So one heater acts as a backup for the other. We have 8 lights per tank on the systems. And then we have 6 Vortec MP40s per tank. So that means just 18 in total for an entire system so that we can mimic ocean currents. We have media reactors to house active carbon and other medias, depending on what needs to be added or removed from the systems. We have circulation pumps, one for the main water supply and other one supplies oxygen water for the protein skimmers. So all of these are working in tandem on our systems. And I'm just going to show you some pictures of what I'm talking about. So the Radion lights, most of these items you can get from bulk reef supply, and that's where a lot of our items come from, just because they have almost everything that we would need. So Radion lights and those ones, they have a full spectrum, but we use the blue spectrum light just because it helps promote coral growth. We have wave makers on each system, heaters. For our spawning systems, we have glass heaters, but we found out that those ones tend to shatter a lot easier than the titanium rod heaters. But all of our temperatures are kept within a specific range for most of the systems. We use Apex to control most of our systems, actually all of them. But for our spawning systems, we are able to control the protein skimmer, all the lights, the main water pumps, all of those are controlled using the Apex system on our spawning systems. Everything else we try to do manually just to make sure that we don't have any faults whenever electricity or power goes down. We have filtration, so biological filtration from the live rocks. We have chemical filtrations from the carbon, and we have mechanical filtration going on in our systems from the filter salts. We have other filters in our systems that are attached to the main pump, just so we can take out as many small particles as possible. And this is our facility. Yes, it is blue. So even the pictures come out as blue. And these are the protein skimmers. This is one row of a system. So we have three identical ones, and all of them have the media reactor. They all have their own separate equipment because we wouldn't want cross-contamination to happen. And all of these are in the closed loop systems. For us, we just need to heat our waters. We use the AC unit as the control, like it acts as a chiller. So on our main holding tanks, we don't have any chillers, neither on the medical tanks. But on the spawning systems, we do, just because those are completely separate. And that's because with our spawning systems, we plan to do something kind of special with them. We plan to take some of the corals that are from our holding tanks, put them in the spawning systems, and then some corals that tend to spawn maybe at midnight, we plan to change the time zone on the spawning system so that it'll be completely separate from the holding tanks. And then instead of spawning during midnight, they're going to spawn at midday, so right at noon. And that's our plan for this year, and hopefully it'll work. We've seen it being done in Florida, but it's time for us to try it here at home. With our coral gene bank, we house 15 different species, and we plan to add more species. But our goal for this year is to have a minimum of 20 colonies of each one of these species. Like I said, we have 3 identical life support systems. We have 3 medical tanks and 2 spawning systems. With the spawning systems, they have a connected settlement tank. So after the coral spawn in their spawning system, we're going to transfer the gametes over to their settlement tank so that they don't have to go into the main holding tanks and just disappear. And some of you might wonder, okay, why do it in the Bahamas? I mean, things are expensive there and you don't have any reef stores. So why here? Why? Because it's our country. We're here because we have a high coral biodiversity. We have reef here. And we need to take care of what we have. We need to take care of our home. So we can just take the corals that we need from our natural environment and house them here so that if there is something that happens out in the ocean, we're able to spawn the corals here and potentially grow those and put them back into the ocean and help to rebuild our reefs. Yes, we understand that we have a limited amount of capacity because we only have so much space, but we're just one strategy that's being used in order to help outplant on our reefs and help to restore our reefs. We're also here just in case we have another bleaching event because about 2 years ago, there was a rapid bleaching event that happened where all the corals they went through an extreme heat wave stress. So we're here, we have one open tank. So just in the event that something does happen to the corals in the wild, we're able to bring in some of the corals from our nursery or from the wild and place them in our tank so that they can be preserved here before they go back into the ocean when things stabilize. We're here to act as a training agency as well. So we want to strengthen our local conservation capacity. We want more coral scientists, not just us because there are just 3 of us and our colleagues who work at HIMSS, but we want to build more Bahamians and empower more Bahamians into becoming interested in corals and coral reef. Most people, they go out, they go to the beach and they enjoy the water and the fish, but corals are there too. Corals are there to help sustain our reefs, our fisheries, and the life that we enjoy in the Bahamas. And all of this takes time and planning, all of this facility. So when it comes to coral collection for us, we have to pre-identify which species we want to take out of the ocean. So I said that we have 15 species. We had to decide which 15 species we wanted to take out, what size they were going to be, and where we would collect them from. So we would use GPS coordinates on Google Maps and then hit those target areas at certain times in order to maximize our collection. But it doesn't always go how we want. So sometimes you would have to pivot, but we try to do things in an ethical and a minimalistic impact approach. So we try to hammer and chisel our corals out of the ocean slowly, and we go there with a plan written out stating which corals, which species, which sizes we want, and we go and look for our target species. We want to get corals from many different areas around the Bahamas just because we want to increase that coral biodiversity. We don't know what genetics these corals might have. They might have higher heat tolerances or be hardier. We don't know, but housing them here, we're able to collect samples so that scientists could be able to test them so that they can get a better understanding of what's in our oceans, especially for corals that have already been exposed to stony coral tissue loss disease. That if scientists want to test those, then here would be a perfect example of where you can potentially get samples to do that kind of experimentation. But for coral collection and planning, it takes a lot of planning between teams. So you would not just have persons who are working in the lab to collect the corals. You need persons who are working in the water. You need persons who are running the boats and staff who are working on liveaboard missions, because for some of our coral collection trips, we have been doing coral liveaboard trips so that we can spend more time out in the ocean searching for what we need, not just having to waste gas coming back. So all of that takes time and planning. So far, we've collected from Exuma, Andros, Abaco, and New Providence, different areas around New Providence. And we're still in our coral collection phase. So we're about at almost 2/3 capacity, and we still have more corals that we need to collect. So this is just a picture of what I mean when I say that we have to stabilize the corals. So we go out there and we collect different fragments and whole colonies of corals. The larger whole colonies, they're going to be our brood size corals. And those ones we were able to put in our spawning systems. We have detailed documentation. So when we're in the ocean, we take notes of what corals we're collecting, what depth we collect them at, what measurements they're at, and what the reef site is. And then afterwards, when we get them to the boat that we're on with the corals, we stabilize them on these tiles and using egg crates in a cooler. We have oxygen and fresh salt water going to them. We also check the water quality parameters on each one of these coolers constantly during the day just to make sure that they're stable. While they're on that boat, we give them food as well during the nighttime and daytime because they still need to be fed just to decrease the stress on them. And we make sure that their water isn't fouled. So this does take a lot of coordination, a lot of planning, a lot of writing detailed reports. And then this is just a picture of us moving the corals from one of the boats onto land. So after we transport them from the ocean, we have a team waiting to collect them on the land most of the time. They're wearing dry clothes, they're not soaking wet, rash guards or in wetsuits. They're there so that we can help to follow our biosecure procedures. So when we're moving them over into these bins that have salt water in them, we use the same salt water that they're in on that boat that we brought them in. It's just a slow process of moving everything. And while that process is happening, we make sure that their temperature is stable, they have enough oxygen, we minimize the transport time just so that they can have less stress as possible. Because corals, yes, they grow slowly, and sometimes they might be having an issue, but you wouldn't see that happening until maybe a month later, or in some cases, if they're extremely stressed, they can bleach out in a matter of hours or days. And we have that team waiting in those dry flows just because in the facility, we don't want any cross-contamination coming into the facility. So with our first collection, we were able to do a large scale collection and then place them in our holding tank, our main holding tank, just because we use that as a quarantine system. With our second collection, we had to put the corals in our medical tanks just because they were coming from a different location and we didn't want that cross-contamination happening. So if they had stony coral tissue loss disease, we wanted to see that they're experiencing problems in those medical tanks where we would be able to deal with it before we place them into our main holding tanks where we have our larger collections. Upon arrival into the gene bank, we also conduct another set of visual health checks, verify the records to make sure that the corals are coming from the specific area that we say that they're coming from, and we just double check everything just to make sure that everything is accurate. And we assign each coral to a separate quarantine system. So if we see that corals came from an area that did not have Schedule D in the area, we would place them in one quarantine system together. And if corals came from an area or a coral colony that was already infected with stony coral tissue loss disease, while we're transporting them, we place them in a separate container. And then even with the quarantine, we place them in their own separate quarantine system. So each one of our quarantine systems, they are individualized and they are separate. They have their own protein skimmers, filtration systems. They're like mini life support holding systems. They're like mini ones of these. They have flow on them as well. And all of this is for us to, we do all of this slowly, get them adjusted to our tanks so that they can have reduced stress happening on them. So we have stress reduction protocols, and most of our protocols we got from the Association of Zoos and Aquariums. So we got our training from there, and we adhere to many of their protocols that they use for the Florida Reef Track Rescue Program. So like I was saying, this is our quarantine, one of our quarantine systems. This was the collection from Abaco. So we had many different species of corals there, but with this one, we place this is about the size of one of our igloo coolers. So we were able to collect that and just put those corals directly into that spawning system, that quarantine system. The other ones went into our smaller quarantine systems just because they did come from areas that had stony coral tissue loss disease. Every day we come in here. So it doesn't matter if it's a holiday or if it's a weekend or if it's a tropical storm, we are here. Well, if it's safe enough, we are here. So we get here at 6 a.m. every day and we do visual inspections on all of our corals. The corals, they act differently. Like every one of them, they have their own different personality. So each one of them, we check for tissue condition, their polyp behavior, and each one of the corals, they act different. So some corals, they prefer to put out their polyps during the night times, but even if the lunar phase is out, they wouldn't want to extend their polyps. Others, they wait until the sun is fully up and then they start waving their tentacles around. You just get to know your corals as you work with them more and more. But if there is something that goes wrong in our tanks, The good thing is that we have cameras around the system so that we can see what's going on, not inside the system, but around the room in the system. And then we can conduct early intervention by coming in early and by looking on the Apex system that we have, or just by checking the cameras. So right here is what it looks like to get an alert from our Apex system. If the pH is out of range, or if the temperature is out of range, we get Apex updates to our phones, to our laptops, whatever the app is on, we get those notifications. And it can happen extremely late at night, as you can see. We test water quality every day. Every day we check the temperature, salinity, pH, alkalinity, calcium, and magnesium, and that happens every day. So we go through a lot of test kits just to make sure that these corals are keeping in a stable range. But once a week, we check a whole host of other nutrients such as ammonia, nitrites, nitrates, phosphates, because we are feeding the corals twice a week. So with all of that feeding, we need to make sure that we're not spiking the harmful elements in the tanks too much. So these are our ideal ranges within our systems. So we try to keep the phosphates at 0.03 ppm, and we try to keep the nitrates and ammonia levels low as well. But every year, we try to do an in-depth testing of the trace elements that are in the systems, because corals, they need not just these things to grow, not just calcium and alkalinity to build their calcium carbonate structure, but they need amino acids, they need all kind of things in order to grow. It's just like your body. You need not just meat, but fruits and vegetables and grains, everything, just so that you can have a nutritious balance. So corals do the same thing. They need a whole host of things. And with the corals, we also conduct our biological monitoring and health assessments. Well, we do small wellness checks every day, but an in-depth wellness assessment every 3 months, along with pictures of all of the corals. So this is just a picture of a coral that was brought in to us, well, that we brought in. This is it a month after, well, the top picture is what it was like when it was brought in after a month period. And then about 6 months later, it's bleached. But for us, we looked closely at it. Well, this coral went through a few changes. So it paled, it colored back up, and then it bleached out again. But throughout all of this, it's still eating. Like, we look closely at it every day. It still extends its polyps, it's still eating, it's still taking in nutrition, and it's starting to color up again. So we just keep an eye on these trends with these corals and keep them in our database. We check their growth rates. So we measure these corals to see if their living tissue is growing, if their skeleton is growing. Check their coloration, their survival, because unfortunately we have lost a few corals. It is stressful to move corals from out of the wild into our systems, but we still do have the vast majority of what we have collected, and we check their health trends as well. So some, they had problems, but then they started to color up again, and they got healthy, or we would have to give them a dip in maybe Lugol's or in Coral Revive or in something else. And they have recovered. So this is just a video of what it's like for us to feed some of our corals. Well, one of our corals, our Mycetophilia. If you look closely, you'll see its mouth actually opening and it's taking in reefroids. So we feed our corals predominantly reefroids. It's from Blackwell Aquatics. We give them polyp booster before we give them food. And for some of the corals that have the larger mouths, we give them a frozen mysis shrimp. So we try a few things as well. So some liquid supplements for our coral systems, revive, not just revive, restore, that's what it's called, as well as zooplanktos, and some other supplements that we have. Corals are mixotrophic feeders, so they not just photosynthesize, but they do go through food as well. So that's why we feed them. And with us feeding them, we hope that it helps them to support their growth and to enhance their resilience. We have a few staff. Well, the main staff in the gene bank are myself, who represents management, as well as our two coral aquarists who are here. So we're here on a weekly basis. Well, we're here every day. And we also have support from our coral technician team, and we have interns. So from this picture, one of our coral aquarists, Stefana, she gave a talk at UB, and two of the students that were there inside the audience, two of the students that were there in the audience, they came and they did an internship with us at the Coral Gene Bank. But she'll talk more about that coming up because this is her passion.
Hello. Okay, there it is. So my background didn't exactly begin with coral husbandry. I didn't grow up dreaming of raising corals, but here at PIMS, leaders like Craig and Valeria have a talent for turning curiosity into capability. From day one, we were encouraged and occasionally thrown in head first to learn everything we could about coral husbandry. Each member of our team was selected to participate in the Coral Aquarist program, a year-long initiative developed by AZA's Florida Reef Track Rescue Project. The program was designed to meet the growing demand for skilled coral aquarists and conservation practitioners using the care of rescued Atlantic coral broodstock and the management of coral gene banks. And that's just one of the many training opportunities PIMS offered. Working here also opened my eyes to the importance of outreach. PIMS has given me the platform to step beyond the tanks and into the classrooms, where I visited countless of schools, and I am currently coordinating presentations to share what we do as coral aquarists. So while we may spend a lot of the time caring for corals behind the scenes, our mission doesn't stop there. We're also committed to spreading the knowledge because the goal is not just healthy corals in a room, but empowered Bahamians equipped to become the next generation of coral conservationists. So just as a side note, as Gimel was mentioning, one of the slides mentioned the interns. One of the very first outreach initiatives was an invitation from Danielle to speak to a small island sustainability class at the University of the Bahamas. During that session, I was introduced to the students to the Coral Gene Bank, and something about the Gene Bank, whether through my presentation or the team's efforts that day, clearly resonated with them. As two of the students, Sia and Kirkwood, they both reached out and have gone to intern with us here in the Gene Bank. It's been a great example of how outreach can give Bahamians the opportunity to gain hands-on experience learning about coral reefs and marine conservation.
Thank you, Steph. So Max isn't able to join us today, but he has been vital in setting up all of our systems. So he just wanted to share some of his experience. So this is just him underneath our tanks, wiring up actually everything. So wiring up our lights and our MP40s and putting together our systems like the medical tanks with the help of Duran from the coral technician team. So from him, he said that I had some training and did a lot of reading on system design and installation itself. But I'm more of a hands-on learner, and this was the best way to cement the knowledge in my brain. Because, well, being able to do this, I know so much more about these systems than I initially thought there was to know, which makes troubleshooting problems a whole lot easier. With all the equipment and materials we need in the room, now Now it was time to start the room and put them in the correct places, putting everything for each system in the correct tank to make putting together more efficient. This part was honestly like playing Tetris. Once everything was in the correct place, it was time to assemble the jigsaw puzzle. This was when we found out that not all the parts can work where they were originally set to. And here came the modifications and changes to make it work where we needed to do it. When you think that you have everything, when you think that you have everything needed, then there was a few items that were not thought of. And when you realize that this is missing, you get a few setbacks, having to wait on things to be shipped in, which felt like forever, especially when you are on schedule. This taught me to work on other things while waiting for something else. The setup itself was a mix of straightforward installation and then a bit more difficult section. I had to learn how to install lights, design the RO and saltwater mixing station, and figure out the installation for the quarantine and spawning systems. Learning how to dial in the pumps and even a bit of coding or programming for the lights, heaters, and the Apex systems. After the installation was done, it was all about now learning the evolution and adaptation of the systems running at first, cycling, the battle with algae, and then adding up the corals. When you think it was done, now that they have been added, there is something else to learn. The addition of corals, we have to make sure water parameters are in the approximate ranges, and these systems have been running for about 7 months with corals. And on a monthly basis, we have to adapt and make changes to our tanks. Feeding too much can cause a spike in nutrients. If you remove too much algae, it can lead to too much nutrients, and then have too much algae in the tank can deplete the nutrients, leading to other problems. And the corals using nutrients, since the corals are using nutrients, we have to calculate how much we need in order to feed the corals. The longer that they are in the system, there will always be changes, and there will be a need for us to adapt to their needs. So he just put together this short video of the corals, the Coral Gene Bank being built and what we started from. So we're coming down to the end, and we do have an ex situ system, just meaning that we do conservation on land, we do aquaculture on land, and that doesn't mean that we're doing this system is any better than any other conservation system out there. We're just another system that can be used in order to integrate coral conservation in our Bahama land. So our system is just one of the tools that can be implemented in order to support restoration, and it helps to inform management of what's happening, and to keep our corals in a safe environment, just in case there's something that happens. We need all of these different things in place, like the coral nurseries, the mangrove restoration, and different aspects of coral conservation. We need all of them working together in order to make our natural environment better. Some challenges that we had with setting up what Max wrote down was a part of it. So the initial setup, it took a lot longer than we thought that it would. And that was partly due to shipping delays and some other things. But we had some environmental stressors of the corals when they first came into the tank. And we had water quality testing problems. So we had to ramp up our water quality. We even bought an instrument called the HAC 3900, D 3900, just so that we can do more in-depth water quality testing in the gene bank. So we wouldn't have to rely on outsourcing that. We still are undergoing electricity and internet stability problems, but those who live in the Bahamas or any small island nation, yeah, that's just what you have to deal with. And funding is still an issue. But some solutions that we have enacted are fragmentation. So some of the corals that we started to lose, that started to see signs of stress and go into shutdown, we fragmented them. So we were able to save some of those genetics on plugs. And I have a picture of them coming up. And most of them are doing fantastic. But another solution that we had, we started bulk ordering. We implemented the robust water quality testing, and we ramped up the feeding, and we tried out different feeds for the corals. And we conducted more wellness assessments and monitoring for our corals. And we reached out to the AZA. They have many persons who are working on that reef track rescue program, who are experts in their fields for coral husbandry. And if we have problems, we reach out to them because they have been at this for many years. So they've been through all of these things, and they have been vital with helping us going through our growing pains. So these are some of the fragments that we were able to save. So before, we just saved like small pieces of some of the the D-labs, the Peasters, a piece of MJAC and CNAT. And this was the small piece of CNAT that we were able to save. It probably just had like 1 or 2 mouths on it. Now this is what it looks like. It's taking over the entire plug. And we've seen growth in all of the other fragments as well. We lost 2 of those, but those were duplicates, but we still have those genotypes. And these corals are growing rapidly, and we're probably able to move them onto their own tile in about a month or 2. Corals grow really slowly, so it's going to take them some time. Looking ahead, we want to expand our collections. We're a small facility, but we want to make sure that we aren't just the first, that we have other facilities around the country. But that's far in the future. We're still trying to finish our facility here and collect what we need. We want to increase capacity and build more coral scientists in the Bahamas. We have partnerships with the government, with UB, and with BAMSI, and we want to strengthen those partnerships. We want students to come and learn from us so that they can get interested and get trained in this niche field. And we want to make sure that we help with all of the national strategies for protecting the environment and working to help our oceans recover from climate change. And that's it for us. Every healthy coral in here represents a future reef. And hopefully when we start spawning, we can help with building that future reef. Thank you all for listening. And if you need to get in contact with me, just feel free to reach out. My email is on this slide.
Thank you so much, Gimel. That was an awesome presentation. Thanks as well, Stefana. So before we get into the Q&A session, we just want to remind everyone about the webinar feedback link. It was shared in the chat, and we would really appreciate it if you fill out that link. So we're just going to get right into the questions. So the first question we have is from Liz. Are there internship opportunities for high school students?
Unfortunately, at this time, we only have intern opportunities for current students of UB and for BAMSI, but you might want to look into the Rising Tides program and chat with Danielle about that.
Yes, so if you are interested for high schoolers to do any sort of work with us, we do have a youth program open right now. It's called the Rising Tides program. Just check out our website. You will see all the information. Again, Rising Tides. Thanks for the plug-in, Gimel. So the next question is, Will all attendees today receive the recording via email? So I think I can answer that one. Yes, you all will receive the recording. Once it is available, the email will go out to you all. Now, during this Q&A session, we really would encourage you guys to send any questions that you may have. Feel free to drop them into the chat, into the Q&A section, or if you would like to verbally ask your question, you can raise your hand and we can see about unmuting you and you can speak to the team person to person. But if there are any more questions, please let us know. We do have a next question. Do the holding systems require additional or addition of calcium, alkalinity, or magnesium?
Yes, so they do. Right now, we don't have a calcium doser on the tanks, but for us, since our tanks are relatively new and our corals are relatively new to the system, the only thing that we've been dosing so far is sodium bicarbonate, just to keep the alkalinity stable. But for other parameters, we're able to just do a water change, and that helps us to just get those parameters up to where they need to be. We haven't needed to dose magnesium as yet, but our corals are still young and most of them are really recent, but we have what we need in order to dose them when we need to dose them.
Okay, awesome. Thank you. So while we wait for further questions, I'm just gonna ask my own personal question. So I know you say they have different personalities, really, really random. So you say the corals have different personalities. Do you have a particular favorite, or is that just a completely unethical question?
I have favorites, but you know, you should not choose favorites when you have babies.
Yeah. Okay.
But I have, well, my favorite species, well, they're agricode, they're distos, but they're my babies. Especially there's this one that it looks weird. It looks like the manta from SpongeBob. Like that's what it's shaped as. So
Oh. And that's your favorite.
that's my personal favorite, but I know that Steph and Max, they have their own favorites. Max, he likes the Mastidophilias, especially when they're opening their mouths and taking in all of that food. Like they are like caverns. They just open up and you're like, okay, where did this gaping hole come from? And then like their tissue is growing really rapidly because they're eating so much. But Steph, do you have a favorite?
No, I like them all.
Okay, we being politically correct. Okay, so we'll move on to the next question. Sani asks, do you adjust your parameters to the local conditions where you get the corals from?
Yes, we do. So, well, we try to do like a general area parameter set. So for most of the water that we tested out in the ocean where we were, they had like a certain range. So we just keep them at that range. Like the waters in New Providence were similar to the ones in Apico and in Andros. And we use an app, like a table online to tell us when the sunrise and sunset is. So we follow what's going on outside, as well as the water temperatures with that as well.
Okay, so Alberto is asking, are there any job opportunities open at the moment, or is there a way to stay updated on that?
We have no job openings at the moment, but you can always check on the Perry Institute's website to see if there are any job openings in the future.
Awesome, thank you. Nancy asks, what database slash record system do you use to track animals in the lab? And is the same system used to track the animals in the wild?
Currently, our gene bank, we just do it rudimentary style. So we use Excel sheets, Google Sheets, and we take pictures of everything, and then I manually separate each one and put them in their own folder. So I am the sorting system right now, but for tracking them in the wild, the coral technician team, they use a wide variety of things. So they do photogrammetry, they do tagging of the coral sites, just so that they can know which areas are which. We use GPS tag locations, and photo mosaics of the area of that coral reef.
Okay, awesome. Thank you. So Daly is asking, what is permitting like to collect wild corals? Was it a difficult process?
Permitting it's a process. And I understand why it has to be rigorous, because these animals, they are endangered. And many of them, they're very sensitive and they're very vital to the Bahamian economy. So it is important for our government to take rigorous action to make sure that who's taking the corals out of the ocean, that they're skilled to do so. So it's a lengthy process and they work along with us to get that done, but it does take some time.
Okay, understandably so. Thanks for that answer, Gimel. Just going to check for some more questions. I think we have time for one or two more. So if you guys have any last minute questions, please feel free to drop it in the chat. Okay, so we are getting a whole lot of thank yous for the presentation, Gimel. But I don't think we have any more questions. Oh, sorry, one more question. How can people support the work of the Gene Bank and coral conservation in the Bahamas?
Donate. That's what we need. We need donations, and then if we ever need assistance with collections in the future, there might be an opportunity for others to lend a hand. But to support the Gene Bank, just donate. That's primarily it for now.
Awesome. Another question. Do you work with gametes or taking advantage of the spawning season?
Sorry, can you say that again? My internet dipped out.
No problem. Do you work with gametes or taking advantage of the spawning season?
That's the plan for this year. So we haven't done so as yet. That's why we have the spawning system. So hopefully we'll be able to collect some gametes this year. We just opened up in May of last year. Not even in May, actually in June. So we missed the spawning season last year. So we're going to try and do it this year.
Awesome, very exciting. Okay, next question is, I know Hawaii has regulations on the types of sunscreens they allow. Does the Bahamas have anything like that?
Unfortunately, no. That's it. Yeah.
Okay.
Sad.
Sad face. Okay, we do have time for more questions. So
Well,
I think, sorry?
No, I was just saying thank you to everyone because I keep seeing the thank you messages.
Yeah, y'all did a wonderful job. But I think that may be the last of the questions. So with that, we will close out. So thank you again, everyone, so much for joining us today to learn more about the Wild Reef to Ex Situ system, bridging aquaculture and conservation. Also, huge thank you to Gimel and Stefana for this very informative presentation. For more information, please visit our website at perryinstitute.org and also follow us on our social media channels at Perry Institute for Marine Science, including Facebook, Instagram, Twitter, LinkedIn, as well as TikTok. We hope you will join us again for our future webinars. Thank you again, and please enjoy the rest of your day. Bye, everyone.